Welcome to the fascinating world of complex carbohydrates. Today, we're going to explore polysaccharides and glycoconjugates. These are not just simple sugars, they are massive, intricate structures that serve as the unsung heroes of your body. By the end of this lesson, you will understand how cells store energy, build their structural frameworks, and communicate with one another. Let us dive in. To understand these massive molecules, think of a single simple sugar, or monosaccharide, as a single Lego brick. On its own, it is quite simple. But if you snap thousands of these bricks together, you can build a massive castle or a long bridge. In biology, that massive, complex structure is called a polysaccharide. Now, imagine you want to attach your Lego structure to a completely different toy, like a wooden block or a metal car, to create a hybrid toy with special features. In the biological world, attaching these sugar chains to proteins or lipids creates what we call glycoconjugates. Now that we have our Lego analogy in mind, let us look at the formal definitions. Polysaccharides, which are also known as glycans, are large polymeric macromolecules. They are composed of many monosaccharide units linked together by specific connections called glycosidic bonds. These structures can be linear, resembling a straight chain of beads, or they can be highly branched, looking much like a complex tree. When a polysaccharide is built from only one type of sugar monomer, we call it a homopolysaccharide. Conversely, if the chain contains two or more different types of sugar monomers, it is known as a heteropolysaccharide. Building on the idea of hybrid molecules, let us explore glycoconjugates. These are complex, biologically active molecules where one or more carbohydrate chains are covalently linked to a non-carbohydrate moiety. This non-sugar part is usually a protein or a lipid. There are three main classes you need to know. First, we have glycoproteins, which are proteins with oligosaccharide chains attached. Second, we have proteoglycans, a specific subclass of heavily glycosylated proteins. Finally, we have glycolipids, where carbohydrates are attached to lipids. Each of these classes serves critical roles in cellular communication, structural support, and immune responses throughout the body. You might be wondering why these molecules matter so much in our daily lives. The answer lies in their incredible biological functions. Without polysaccharides like glycogen, your muscles and brain would run out of energy within minutes during a workout. Glycogen acts as your primary short-term energy storage. In the plant kingdom, the polysaccharide cellulose provides the rigid structure that allows trees to grow hundreds of feet tall. Glycoconjugates are equally vital. For instance, your specific blood type is determined entirely by the glycoconjugates present on the surface of your red blood cells. Furthermore, the slippery mucus protecting your stomach from its own acid is packed with heavily glycosylated proteins. Having discussed their functions, let us visualize what these molecules actually look like. On the left side of this diagram, you see a homopolysaccharide. Notice how it is a simple, repeating chain of identical blue hexagons, much like amylose. In the middle, we have a heteropolysaccharide, which features a branched structure made of different colored hexagons, representing different types of sugar monomers. Finally, on the right, look at the glycoconjugate. The green ribbon represents a protein backbone, and the attached multicolored chains are the oligosaccharides. This clearly shows how carbohydrates act as massive structural components added onto proteins, rather than just tiny tags. Before we calculate the mass of these massive chains, we must understand the chemistry of how they link together. When two monosaccharides join to form a glycosidic bond, they undergo a condensation reaction. This means that for every bond created between two sugars, exactly one molecule of water is removed and lost. Consequently, the effective mass of each sugar residue inside the chain is its original mass minus the mass of that lost water. It is also crucial to remember that a chain made of N units will only have N minus 1 bonds, meaning the very ends of the chain do not lose that final water molecule. This chemical reality leads us directly to the formula for calculating the total mass of a polysaccharide. The formula states that the total molecular weight equals the degree of polymerization times the difference between the monomer weight and the water weight plus the weight of one water molecule. Let us break down these symbols. MW poly is the total mass of the final chain. dP stands for the degree of polymerization, which is simply the total number of sugar building blocks. MW mono is the mass of a single free sugar unit. MW water is the mass of water, which is 18 grams per mole. We subtract the water weight for every unit and then add one water molecule back at the end to account for the unlinked terminal ends. If the degree of polymerization increases, the total mass increases proportionally. Now that we have our formula, let us apply it to a real example. We want to calculate the molecular weight of an amylose chain, which is a plant starch. We are given that the chain has a degree of polymerization of 1,000. The mass of a single free glucose molecule is 180 grams per mole, and water is 18 grams per mole. First, we substitute these values into our equation. We calculate the effective residue weight by subtracting 18 from 180, giving us 162 grams per mole. We multiply this by the 1,000 units, yielding 162,000. Finally, we add back the 18 for the terminal ends. Our final answer is 162,018 grams per mole. This shows how efficiently plants pack away massive amounts of glucose. Moving from pure polysaccharides to hybrid molecules, we often need to know exactly how much of a glycoconjugate is actually made of sugar. Conceptually, we just find the mass of the sugar portion and divide it by the total mass. The formula shown here does exactly that. The percentage of carbohydrate equals the total mass minus the protein mass, all divided by the total mass, multiplied by 100. In this equation, MW total is the weight of the entire hybrid molecule. MW protein is the weight of just the naked protein backbone. The numerator represents the absolute mass of the attached sugar chains. As the number of attached sugar chains increases, the carbohydrate percentage will naturally increase, changing the physical properties of the molecule. Let us see this formula in action with erythropoietin, or EPO, a vital hormone for red blood cell production. We are given the total molecular weight of 30,400 daltons and the protein backbone weight of 18,000 daltons. We want to find the carbohydrate mass fraction. First, we subtract the protein mass from the total mass. 30,400 minus 18,000 gives us 12,400 daltons. This is the absolute mass of the sugar. Next, we divide this sugar mass by the total mass of 30,400 and multiply by 100 to get a percentage. The final answer is approximately 40.8%. Physically, this means nearly half of this crucial hormone is made of sugar, proving that carbohydrates are massive structural components, not just tiny tags. We have covered a lot of ground today, so let us review the most important ideas. First, polysaccharides are massive chains of simple sugars that serve as the heavy-duty structural materials and energy vaults of the biological world. Second, remember the chemistry. Linking these sugars involves a condensation reaction that removes water, which is critical when calculating their massive molecular weights. Third, glycoconjugates take these sugar chains and attach them to proteins or lipids, creating highly specialized hybrid molecules. Finally, as we saw with the EPO hormone, the carbohydrate portion is often a massive fraction of the molecule's total weight, dictating its stability and function. You now have a solid understanding of these fundamental biomolecules. Great job!